I have a confession to make. My name is Donovan Bailey, and compared to most of the other speakers here, I know very little bioinformatics. And so I feel much better having made that announcement. So why am I here? I believe I'm here, according to Jason anyway, because I represent at least one important demographic that is really the target of iPlant and why iPlant is, was conceived in part. And that is for people who don't necessarily have the skills or the resources or all the computational tools to be able to do this. And I'm certainly in that group. My laboratory have been working for about 10 years on this group of legumes that are sort of unimportant to you right now, but the genus is called Leucaena. And we've been going through trying to understand the evolutionary history, understand hybridization, a number of different things in this group. And we did it through good old-fashioned heavy-duty PCR, large numbers of low-copy nuclear genes, cloning. We spent all of our time in the lab and very little time in front of the computer. And as we started to go forward with publication on this, I looked at my lab and we were really proud of what we'd accomplished, and I smiled and I said something like, we've accomplished something great here with what's a dinosaur technology. We really need to be able to move on and do some other things, but we didn't have the expertise to do it. And so I attended one of these wonderful workshops actually run by Jason, and that really gave me a window into even the kinds of tools I needed to become familiar with that I wasn't familiar with before. And that was mostly, for me, the discovery environment, to be able to start using those tools. And so I had another opportunity, which was to use that first experience with iPlant as a springboard onto some other resources as well. And in this case, I was able to take a sabbatical and go to Oregon State University. Now, most of the primary work in my lab at the moment focuses on a transcriptome sequencing project and a bit of a genome sequencing project that I'm not going to talk to you about. But one of the first pieces of data we got from the beginning of that project was something really simple, and that, well, simple in some respects, and that's a chloroplast genome. Yes, we know a lot about legume plastomes and chloroplast genomes. There's 11 of them sequenced, so why do more? Well, the legume family actually shows a fair bit more atypical variation among their plastome genomes than most other angiosperm families, which are pretty homogeneous on average. And so they have all sorts of major inversions, changes in gene shifts to the nucleus, lots of weird things, so we need to know more. But when we start comparing all of our different rosid plastomes here, by their total size, there's a couple of interesting things to consider. First of all, some of the largest rosid plastome is Pelargonium hortorum, a hybrid, and it's at about 217,000 bases, so it's a pretty big plastid genome, and that involves a large expansion of an inverted repeat. If we look down this list and look for, well, who are the next three? It's all three of our Mimosoid plastomes. So these are actually pretty big. We can look into these sub-regions within those plastomes to try to see what's going on, perhaps, or get a clue. And we can see that Leucaena there in the middle has the largest large single copy region of any of these. And the other two or Mimosoid legumes are also quite large. They're at number 2 and number 3 on this list as well. Now, Inga also has something else going on in terms of its genome expansion, and that is it has a larger inverted repeat than any other legume that actually has an inverted repeat. So there's more than one thing going on here. So we have a clue that tandem repeats are a significant part of this, and we wanted to look at their distribution. So we map them here. Each one of these little green dots, or pardon me, blue dots, is a tandem repeat. They're not proportional to scale, they're just markers here. And you can see that they're primarily concentrated in a large single copy, both in terms of number and in terms of distribution. So this really does account for much of the genome size variation in the large single copy region for 2 of the 3 legumes, 2 of the 3 mimosoids. However, in the third, not only does it have that expansion in terms of the large single copy, but I mentioned it also had a larger inverted repeat. And so what we have here is Leucaena on the right at 164 kb, Inga on the left at 175 kb. And again, the Leucaena has the typical, they both have the typical large single copy inverted repeat, small single copy inverted repeat. And typically this small single copy region has around 13 genes in it. But the Inga plastome has, the IRA of the Inga plastome has expanded itself out to incorporate 9 of those 13 genes. There's only 4 genes left in the small single copy. And so that actually accounts for the other 10 kb in size variation for that particular genome. So through this, we're able to understand a little bit more about some of the genome variation among these different legumes. And again, there's more atypical variation than was predicted previously. So in conclusion, iPlant resources and training in my case really were a springboard into being able to start doing comparative analyses on a scale that was just seemed impossible to me 4 years ago. And so as an outsider in this particular group, I'd really like to say thank you to all the members who conceived of and implemented iPlant and continue to implement iPlant as they pull all these different pieces together. It really is important to an awful lot of us out there.